So it's always, it looks like it is intimidating to give a talk right after Chris' talk. He's like a superstar in New Jersey. The spawn model is the only argument that I make when people. Oh, this is not on. So the spawn model is the only argument we make and we have when people are skeptical about where are we going. So this is where we stopped yesterday, and today I'm going to cover the bottom part, and I'm going to essentially describe a robot that we built both in terms of hardware and software. And the argument I want to make is that our goal is to develop spiking neural nets that have some kind of biological constraints and use these models as controllers for autonomous robots. And there seems to be a spectrum of brain-related algorithms, and I'm using the word related to cover both brain-inspired, when we imitate the learning rules and some computational principles, and the other end of that range is brain mimicking, where in addition to learning rules and principles in the brain, we imitate the structure. And you will see that if we imitate the structure, we need almost no training. So to convince you about that, we're going to play a small game, a small brain experiment. So what I'm going to ask you to do is to read this sentence. Can you read this? And we're going to do two tasks. The first task, as you will be reading it, you will be moving your head. And in the second task, I will try to find a way to actually do the same thing for you, although you will be keeping your head stable. Are you ready? So experiment 1, can you read this? Yes? No? Yes. So the second experiment is this. Can you read that now? Someone might say that it's the sampling frequency of the projector. No, it's not that. It's, in fact, this is what your eye is doing. Deep inside your brain, there are canals. Inside them, there's fluid. And inside this fluid, there are hairs that are embedded. And whenever you move, due to inertia, the fluid moves. And without these hairs that are actually neurons that project directly to motor neurons in your eye. Believe it or not, this pathway from sensing to motor takes 2 neurons. And this is an example of a way that if you follow the structure, you might end up with a model, a spiking neural network, that is a model of a robotic head that we have here. That requires no learning. Now, when we show that to computer scientists, they think of us as imposters. They tell us, well, there is no way that you can build a network, be it spiking or non-spiking, without learning. When we show that to neuroscientists, they think of us as imposters because they say, so what? I mean, yeah, it makes sense, right? Since I don't have much time, what I want to focus here is the second component of that autonomous robot, which is essentially a controller that helps that robot to orient itself in an unknown environment. And we're using the biological navigation system that started back in the 70s. And since then, there are many cells that have been associated to this biological navigation system, starting from the place cells that were discovered by O'Keefe in the 70s and going all the way to the grid cells and other cells. So what we did is we abstracted away this network. And the problem with that is that these cells have been experimentally found. So we know that they are activated when the robot, our brain is doing the navigational task. But what we do not know is their connectome. We do not know their connectivity. So this is why this controller, the spiking neural net, is more at the brain-inspired level and less at the brain mimicking. So we kind of infer about the connections. So what we have here is a robot that its main scope is to explore the unknown environment and keep a memory of that environment. And to do that, I'm going to describe two main principles that we're using. One of them is a dendritic multiplication. So take, for example, this goal cell here. This goal cell will be activated when a goal has been identified. And how do you identify goal? In our case, we need to have a green, blue, black wall, and again, a wall. So what you need is to have a concurrent activation of a neuron that tells you that you are in a border, and another neuron that tells you that I found that green or blue target. And that concurrent activation makes that goal cell to be activated. Now, how do you memorize that? When you have that goal being activated, and one of these place cells that I'm going to describe next how we generate them, being activated through STDP, you increase these connections. So what that means is that if for any reason you turn off the lights or your sensor doesn't work, and this pathway here is not anymore valid, or you cannot trust, or you do not want to trust it anymore, the STDP will take care of that. So you have a memory of the unknown environment stored in these connections between the hippocampus and the prefrontal cortex, the simulated parts of it. This is another component of our model. What we wanted to do is to assume that the environment is unknown. So we need to transfer the knowledge from the egocentric mode, so the mode that I have right now, to the allocentric. So if I would have a bird's eye view of the entire environment, and to do that, we use, again, dendritic multiplication using the head direction cells, which are another cell that is associated with the navigation system. And this is the last result slide. So it shows a very simple motor planning, local planning. So what we have is we have 4 different cells that tell to the robot whether it should move to north, south, west, or east. And what is happening here is that these border cells are actually inhibiting the cells that should not be followed. And this makes that robot not to fall into the walls. This is a slide I owe you since 2017. I didn't make it to the NICE 2017 conference. And this shows how we built the place cells, the head direction cells, using Bayesian inference that I would be happy to discuss offline. And this is, again, from last year. We had some progress since then. And this shows the main cells that are associated with the spatial navigation system. And the neuro theory exists means when there is a neuroscience paper that actually describes the connections between these cells. And the dotted lines are to note that we do not know whether there is a connection or not. So we have to create a model that either infers or not. About benchmarks, we had this discussion yesterday. I would say that the word "it works" is sometimes the only benchmark that for now we're satisfied, given that this is a new direction we are taking. And in the end, this is the overall work that we do in the Combra lab, and these are the people behind it. And I would love to have any questions that I would, hopefully try to answer.